I'm super excited to be able to show some of my work today, which focuses on how alternative translation shapes our complex proteome. And so I wanted to start off by showing you how many of us draw a cell as kind of this simplistic blob over here. And honestly, this is pretty good. Sometimes people will just draw a circle and call it a day. And maybe if you're a hardcore cell biologist, you'll maybe draw a cell like this, but even then, this doesn't fully represent the true complexity of our cells, which are really filled with thousands of macromolecular machines that need to coordinate so many biological processes. And so during my PhD, I've been fascinated by the cellular complexity, and I was really interested in asking the question of how do our cells achieve this remarkable complexity? And so the answer lies not just in the number of genes that we have, but how we can decode a single gene into multiple protein products. And so many of you guys are aware of alternative splicing as a mechanism, but today I'm going to talk about alternative translation. So translation usually starts off with the ribosome scanning the RNA, eventually hitting a start codon, typically AUG, where the 6DS subunit will join and start making a protein. So what we know in Ensembl or RefSeq or your favorite reference databases is known as the annotated protein. But in some cases, the ribosome will scan past this annotated start codon, starting downstream to make an N-terminally truncated protein isoform. Similarly, the ribosome can sometimes start in the 5' UTR, typically at a non-AUG start codon, to generate an extended protein product. So which genes have alternative start sites, and how do we even identify them? And so this was really made possible through the development of translation initiation site sequencing, which relies on two drugs to stall the ribosome at the start codon. You can then treat your lysate with nuclease and sequence the fragment of RNA protected by the ribosome. And so this should really map to start codons. And so using this approach along with computational modeling, I was able to identify these alternative start codons similar to what was reported in the literature. What I want to particularly highlight over here is the large number of these alternative N-terminal isoforms, or what I'm going to refer to as the dark proteome. For a while, this was discounted as translational noise. But today, I want to go into some of the secrets lying within this dark proteome. In the first part of my talk, I'm going to start downstream and highlight just how important these alternative N-terminal protein isoforms are for a core cell biological process, tRNA biogenesis. I then also leverage this to understand aspects of rare diseases. In the second part of my talk, I'll go upstream, and I'll talk about how alternative translation is dynamically rewired across the mammalian cell cycle. So I want to start off by going through one of my favorite genes, TRNT1. Here I'm showing you the ribosome profiling trace for TRNT1, where I've been able to identify two start sites. This orange one over here represents ribosomes stalled at the annotated start site, and this purple one over here indicates ribosomes stalled at the alternative start site, generating a 30 amino acid N-terminal truncation. So my first question was, well, what are these two different isoforms doing? And so when I took the cDNA of TRNT1, I plopped the GFP at its C-terminus, you get this beautiful dual localization to the nucleus and the mitochondria. But how does a single RNA produce dual localized proteins? And so I showed that this was through alternative translation of TRNT1, and I was able to deconvolve this. Annotated TRNT1 only localizes to the mitochondria, and truncated TRNT1 only localizes to the nucleus. So this localization actually makes a lot of sense given what this gene does. TRNT1 is a gene involved in tRNA biogenesis. We have tRNAs, of course, encoded in our nuclear genome, but we also have tRNAs encoded in our mitochondrial genome. So you really need alternative translation of TRNT1 to process tRNAs in these distinct locations. And so next, I wanted to leverage alternative translation of TRNT1 to understand aspects of its disease. So TRNT1 is mutated in this super rare disease called sideroblastic anemia with B-cell immunodeficiencies, periodic fever, and developmental delays. I found two patients that had mutations affecting only one isoform of the gene. So patient 1 has this missense mutation over here. Most people will just assume that a missense mutation is a complete loss of gene function. But actually, in this context, this frameshift would only eliminate the mitochondrial isoform of TRNT1 leaving the nuclear isoform intact because its start codon is downstream of the mutation. Indeed, that's exactly what I see. Patient 2 has this sense mutation, methionine 30 to leucine. Okay, that's a pretty conservative change. It's also in this floppy region of the protein. But actually, this is also the second start site of TRNT1, and I predict would eliminate the translation of the nuclear isoform. And indeed, that's exactly what I saw. So I thought this was pretty cool, but what gave me even more joy was when I was talking to the clinicians about these patients. So standard SIFD patients typically have mutations that affect both isoforms of TRNT1, and it's pretty dramatic, it's pretty severe. These two patients were outliers. These two patients only had mild and partial symptoms. And so we're going to propose that these mild symptoms in these patients are because they have alleles affecting only one isoform of the gene rather than both. Okay, so just to summarize, I think considering alternative start codon selection is really important when analyzing the consequence of disease alleles. In work that I don't have time to talk about today, I show that this is not true just for TRNT1, but for hundreds of other genes. All right, and so clearly I think that understanding the downstream functions of alternative translation is important, but I think there's even more to be learned looking upstream. The beauty of alternative translation is that it allows you to tune which protein isoforms are made across conditions from just one single mRNA. So in the next part, I'm going to talk about how alternative translation is changing across the mammalian cell cycle where the proteome needs to be dynamically remodeled. And so to do this, I perform translation initiation site sequencing in each stage of the cell cycle, and I'm going to focus on this really dramatic change between interphase and mitosis. Shown over here in this volcano plot is the change in efficiency of each start codon between these two stages, and you can see that there are thousands of initiation sites that change between interphase and mitosis. What was even more satisfying was the underlying mechanism. I showed that this can largely be explained by the start codon identity. This is almost the exact same volcano plot, except non-AUG start sites are now in purple and AUG start sites are in orange. What I hope you guys can appreciate over here is that non-AUG start sites are preferentially repressed in mitosis relative to AUG start sites. And so these data really suggests that there's some switch as the cells enter mitosis that's causing the preferential repression of these weak non-AUG start sites. And so I hypothesized that there was probably a protein that interacts more or less with the initiating 40S ribosome that can explain this change. And so to identify this protein, I purified the 40S ribosome from interphase and mitotic cells, and I used quantitative proteomics to compare interactions. Two proteins really stood out. EIF1 preferentially interact with the ribosome during mitosis, and EIF5 less. I was super happy when I saw EIF1 and 5 come out of this analysis because these are the two key and direct regulators of start codon selection. EIF1 represses the translation of these weak non-AUG start sites, whereas EIF5 promotes the usage of them. And so this lined up perfectly with what I saw in mitosis, where EIF1 is able to interact more with the ribosome during mitosis, and we saw preferential repression of non-AUG start sites. And so next, I really wanted to understand how EIF1 was able to interact more with the ribosome during mitosis. And so unlike most cell cycle dependent changes, the answer was not phosphorylation or expression levels, but it was actually subcellular localization. Most translation factors will localize strictly to the cytosol. That makes sense, that's where translation happens. EIF1 is different. EIF1 has, of course, cytosolic localization, but it also has this nuclear localization. And so I think this nuclear localization can really help explain its differential association with the ribosome during mitosis. When you think of a nuclear protein interphase, well, it's in the nucleus. But when the cells enter mitosis, the nuclear envelope breaks down, releasing the nuclear fraction into the cytosol where it can interact with cytosolic proteins. And so I was able to show by live imaging that nuclear EIF1 is released into the cytosol soon after mitotic entry. I next wanted to test and show that nuclear EIF1 is responsible for this change in start codon selection across the cell cycle. And to do this, I developed luciferase reporter that can measure non-AUG compared to AUG translation. And in control cells, you get preferential repression of non-AUG start sites during mitosis. But in cells where I've selectively eliminated nuclear EIF1, this change is largely suppressed. So these data really supports a model whereby nuclear EIF1 causes this change in start codon selection during mitosis. And so in work that I don't have time to talk about today, I show that this mechanism is even important. And mitotic cells die soon after mitotic entry when cells don't have nuclear EIF1. And so just to summarize, I think that when cells enter mitosis, the nuclear fraction of EIF1 is released into the cytosol, where it can preferentially associate with the cytosolic ribosome, causing the repression of weak initiation sites. I go on to show that this mechanism is actually important for standard mitotic cell physiology. Okay, so just to thank everyone involved, huge thanks to everyone in the Cheeseman lab, particularly Iain for being really the best mentor ever. Huge thanks to Dave Bartel and his lab for all of their collaborations, Whitehead, MIT, funding, and thank you all for having me here.